Welcome to everyone who's tuning in to this session. We at Vitalis Phage Therapy are very excited to start this series of discussions called All About Phage Therapy, where every week an eminent phage scientist from around the world would be joining us to share their experience and their perspective in the world of phage therapy and phage research. To start off this series, we have Professor Sanjay Chhibber, who is joining us from Panjab University, Chandigarh. He is a senior microbiologist at Panjab University and president of the Society for Bacteriophage Research and Therapy in India. He is one of the most experienced phage scientists in India, and his work has been very well recognized in the area of bacteriophage research. He joins us from his home in Chandigarh in the northern part of India. Welcome, sir.
Yeah. Hello, everyone.
I hope, sir, you and your family are doing well and staying safe during these times of the pandemic.
Yeah, that is true. We are very safe and abiding by all the rules and regulations and feeling very comfortable. Yes.
That's good to know, sir.
Thank you. Thank you. Yeah.
So to start off our discussion, sir, I'd like to ask you about about your start in the area of phage research. How did you start off in this world of phage research? How did you get interested in bacteriophages?
The antibiotics, they have been the drug of choice for treating bacterial infections since these were discovered. But in the recent years, the emergence of drug-resistant strains to the commonly used antibiotics has become a problem, and it is causing a major havoc on the health. There are various reasons for this. The number one is that it takes longer to heal when there is an infection with a resistant strain. It prolongs the hospital stay, which leads to higher medical costs. It also has a higher mortality. And therefore, it has been predicted that these drug-resistant strains will be responsible for something like 10 million deaths annually all over the world by 2015. And the reason for the emergence of drug resistance has been attributed to the rampant usage of the antibiotics. WHO in 2014 also recognized that the antimicrobial resistance, that is the AMR, is one of the problems being faced by the world these days because it is happening everywhere. So when I was somewhere in 2006 or 2007, I read an article where it was shown that bacteriophages can be used as therapeutic agents. And this made me think about working on bacteriophages. And it was around that time I started working. However, at that time, there were very few people who were working in this area. But I started my work, and today I have almost published around more than 45 papers in good international journal. The work has been carried out by my PhD students who have got the degrees, and they are well settled now, and some of them are still pursuing their degrees. So the phage work has been done over the years, and I think I have been able to show that this could be one of the alternate strategy because people are looking for alternate strategies to curb the antimicrobial resistant bacteria, and various alternate strategies are being looked into. Of course, we are also working on some of the other antimicrobial alternate strategies, but bacteriophage still remains the one which is getting a lot of attention of the scientists the world over, and this has rekindled the hope that possibly we will be able to use bacteriophages as therapeutic agents. I would like to mention here that this is not a new thing which has come to the mind of the scientists, because the bacteriophages were very much being used as therapeutic agents in the pre-antibiotic era. But with the development of antibiotics, the Western world shifted towards the antibiotics. but there were certain countries like Russia and the Eastern Europe which still kept on working with bacteriophages. And the drug resistance has brought us to our attention towards these bacteriophages once again. So this is the reason that made me work on bacteriophages, and I think that's it.
So the burden of AMR, and when you came to know about the increasing burden, that is what drove you into this area of research.
Yes, yes.
And does that continue to be the driving force for the work that you're doing now? And can you please tell us a little bit about the work that you're doing now in the area of phage research?
Yeah, as I said, when I started working, there used to be drug-resistant strains, but it was not a very big problem at that time. But now we know that these bacteria, they have become even resistant to the antibiotics, which were considered as an alternate to the alternate for the drug-resistant strains. So majority of the strains are now showing heavy resistance towards the antibiotics. So I think the work which we are doing is in this direction only. And as you asked me about my phage work, I would like to say that yes, I'm going to summarize my work in say around 5 to 7 minutes. But if you look at, there were many problems, there were many hurdles when we were working with these phages, and which we have overcome over the years. And today we have preparations which can be used for treating the drug-resistant infections. And to begin with, I would like to say that the first thing was to isolate, purify, and characterize these phages. And for this, we worked on this because characterization has always been an issue. Because in the pre-antibiotic era, one of the reasons for its failure was that time people did not characterize the phages. But now the techniques are available on the basis of which these can be well characterized. And so we have isolated our isolates from the environment. We have purified them in the laboratory, and then we have done the characterization by using the latest techniques, on the basis of which we could identify a number of bacteriophages. We could isolate a number of bacteriophages, and the organisms for which we have attempted to isolate the bacteriophages are Klebsiella pneumoniae, Pseudomonas aeruginosa, and Staphylococcus aureus, both methicillin resistant and methicillin sensitive. And as we all know that all the three organisms are the predominant nosocomial pathogens. So therefore, the majority of the drug resistance has been observed among these nosocomial pathogens. And therefore, we started working on these three organisms. So once the phages were isolated, they were then checked for their co-susceptibility range. The other characteristics, that is the adsorption rate, the burst size, and other things, and the ones which got fit into our strategy. And one thing I would like to mention, that we all know there are two kinds of phages, the temperate and the lytic. But when we are talking about the therapy, we talk about the lytic phages. So we isolated these lytic phages, we checked their host susceptibility range and the other properties, and then we selected a couple of those phages which we thought we will check them in for their efficacy in the in vivo system also. So after doing our studies in vitro conditions, we established the animal models in the laboratory. The mice was used as the model, and we have created the respiratory infections, the pneumonia model, the septicemia model, and the burn wound infection model. And the efficacy of these phages was then tested in these animal models. And we used the whole phages to treat the infections. So the infection was first created in the animals, then we treated them with the phage preparations, And first of all, we initially started with the monotherapy, where the single phage was given, and we got very good results. But as we see that there is always a problem of the mutant generation, so we shifted to the cocktail, using the cocktail of the phages. The only problem with the cocktail of phages is that you have to see the compatibility among the various phages as well. So those conditions were standardized, and then we started their efficacy, the cocktail bacteriophage efficacy in treating the tubercle model, and that turned out to be much better than what it was when we used in the monotherapy itself. And the second thing I would like to say is, even in the animal model, a lot of standardization was needed. The bacteriophage dose was to be standardized. The time of administration of phage therapy, the route of administration of phage therapy, and all this was standardized, and this is published work. And we could use these models, and we are still using these models for checking the efficacy of the bacteriophages. So now, this was when we use the phage as such. But the other condition was where we use the bacteriophage products. For example, the depolymerase enzyme and the endolysins. So first of all, I'll talk about the depolymerase enzyme. When we were isolating the bacteriophages, we saw there was a double halo around the plaques, and we thought of what it could be. And when we looked into, we found that there are certain bacteriophages which form the depolymerase, and there are non-depolymerase producing phages as well. So we isolated these depolymerase producing phages. We grew them at a higher scale. We isolated the enzyme, and since we know this is an hydrolase enzyme, it acts on the extracellular polysaccharide, and therefore we could use this depolymerase enzyme, number one, to destroy the capsule of the bacteria. And here we got good results, because the antibiotic resistance was there when the capsule was present, which might have hindered with the penetration of the antibiotic. But when the organism was treated with depolymerase and then antibiotic, they became susceptible. And this was shown in the animal model in vivo as well. The second place where we use the depolymerase enzyme is the degradation of the biofilms. Biofilms is a community of the bacterial cells which is encased in a matrix, the polysaccharide matrix. It is very difficult to get rid of the bacteria when they are growing in a biofilm mode, vis-a-vis when they are growing in a planktonic stage. And the reason for this is that the biofilm may not allow their penetration into the biofilm of the antibiotic, or possibly it could be because the organisms in a biofilm are generally in a metabolic inert state, whereas some antibiotics need them to be actively metabolically active. So what we did was that when we treated the biofilm, the depolymerase enzyme, we saw that the depolymerase could make a tunnel within the biofilm through which we could deliver the phage, the antibiotic, or the other beta-glytic agents that destroyed the biofilms. So we very successfully were able to get rid of the biofilms in vitro, especially the young biofilms were very, very susceptible. And that was another use of the depolymerase enzyme. The other area in which we have worked is the endolysins. Now, the endolysins are produced by both gram-positive and gram-negative bacteriophages infecting both gram-positive and gram-negative bacteria, but endolysins are more effective in the gram-positive organisms, and therefore using the Staphylococcus aureus and Streptococcus pneumoniae, we have worked with the endolysins also, and these are also, they show good susceptibility to the endolysin. Their efficacy has been demonstrated alone or in conjunction with the antibiotics in the in vivo models. The results have been very, very encouraging, and we have published all this work, yes. And I think this is all about the bacteriophage work being carried out in the laboratory.
So there seems to be a lot of scope to the bacteriophage work that's being done at Panjab University microbiology department. So is it a fairly big team which is working on bacteriophages, and is that a major part of the work of the microbiology department?
No, microbiology is doing a lot of things as far as the research is concerned. Every faculty member has his own area of research, but mine is my area of work is the alternate strategies to curb antimicrobial resistance. And therefore, as I said, I've mentioned about the preliminary work which was carried out in the laboratory, but now with time we have shifted towards developing the what we call the products which could be used for treating the bacterial infections. And in this, I think one product I would like to talk about is the wound dressing, which we have made in the laboratory by using the polyvinyl alcohol and the sodium alginate. And this hybrid hydrogel membrane was for the topical delivery of the bacteriophages and the antibiotics. And this has certain advantages. And these advantages are, for example, this membrane absorbs the exudate due to the strong hydrophilicity, and as a result, it swells when we put it on the wound itself. So it doesn't stick to the wound. So that is a very good point. But at the same time, it delivers the phage as well as the antibiotic at the wound site. Therefore, changing the membrane daily is not painful for the patient, and we are able to then make the wound heal faster as compared to when we have to remove a little bit of tissue while removing the membrane. And we are working, this has been published in the International Journal of Pharmaceutics. This has also been patented, and we are further working on making the better membranes and putting other antimicrobial agents along with the phages so that we have a more effective and rapid killing of the bacterium as far as the burn wound infection is concerned. The second area in which we are working is the delivery systems. What happens is that one of the problems with phage therapy is the neutralization of the phages in the in vivo system, and second is their rapid clearance from the in vivo system to the reticuloendothelial system. And therefore, the need is that the phages should stay in the system over a longer period of time so that they show a better antibacterial potential. We have carried out this work in association with the pharmaceutical department of our university, where we incorporated these bacteriophages into lipophilic systems. For example, the liposomes. Liposomes and containing bacteriophages using the the single phage and the cocktail of phages were created. And again, we used them in the in vivo model, and we got very, very encouraging results. It was much better when we used them without incorporating into the liposomes. But in the skin, we found that the liposomes were not very effective. Why? Because possibly they could not reach the deeper layers of the skin. So for this, we did some modifications, and what is known as transferosomes were created, and which could reach the deeper layers in the skin itself, and they could treat the infection thoroughly, and we could get the good results. The third area, which is again being worked in my laboratory, is the use of endolysin of Streptococcus pneumoniae, the CPL1. And now CPL1 has been shown to be very, very effective antibacterial agent throughout the world. Many laboratories have shown this. But the problem with the CPL1 endolysin is that this has a very short half-life, and therefore this is not possible to be used in the in vivo system. So what we have done is we have prepared the nanoparticles using the chitosan nanoparticles in which the endolysins were incorporated, and then we used this these nanoparticles to treat the pneumonia in case of infection created in mice by Streptococcus pneumoniae, and there were very encouraging results. This is a very good achievement because at the movement there are means to have prophylactically control the Streptococcus pneumoniae infection. But if there is a strain which is resistant to the antibiotics, then there are not many alternatives available. So this system, if it works, this needs to be now go for the clinical trials. It has already been patented, but it now needs to be go for the clinical trials, and then it will be able to we'll be able to say whether this will work in the human beings as well or not. So these are the main areas on which my laboratory is still working on in the department.
Thank you for sharing that, sir. This is really interesting. So we could see in a few years these bandages out in the market as products that people can apply and use as phage-based bandages to fight against burn wounds or skin infections, which are being caused by antibiotic-resistant bacteria. So you mentioned 3 bacteria which you are working on in terms of your work. Can you talk about the common conditions that these 3 bacteria cause as infections? Because a lot of people identify with conditions, may not specifically be able to identify with the bacteria. So there would be a lot of people who might be watching who would wonder, say, Klebsiella or Staphylococcus. Yes, these are bacteria, but what are the common conditions that they cause, infectious conditions? Can you talk a little bit about that?
See, as far as the human system is concerned, all these bacteria, whether they belong to the gram-positive group or the gram-negative group, they are able to thrive into the system and they have the ability to produce the infection. So as far as the bacteriophage therapy is concerned, I feel all the organisms belonging to both gram-positive and gram-negative group are capable of causing the infection, and this has already been shown through. And I feel the only thing is when we are talking about the bacteriophage therapy, then against the gram-negative organisms, I would say the bacteriophage on its own, it works, as well as the depolymerase enzyme works. Whereas in the gram-positives, the phage or the endolysin will work. The reason could be the differences in the cell wall architecture of the gram-positive and the gram-negative bacteria, and that could be the reason that why one is susceptible to the depolymerase and the other to the endolysin. So I think all the pathogens, whether they are gram-positive or gram-negative, can cause infection. And with bacteriophage, both can be tackled if need be. Otherwise, of course, I always say that antibiotics are the mainstay for treating the infections. But bacteriophage therapy will only come when there is a resistance. And bacteriophages can be used on their own. Bacteriophages can also be used in combination with the antibiotics as well. So there is no problem.
So you spoke about how the burden of antibiotic resistance and the growing burden of antibiotic resistance motivated you to start your work in the area of bacteriophage research. This is something which has been highlighted as a problem in the last few years. WHO has been talking about growing problem of AMR. Different countries are also recognizing AMR, and task forces have been set up to combat the problem of antimicrobial resistance. Currently, the entire world is going through a pandemic caused by a microorganism. Do you think that this pandemic will lead to more resources and more focus on the area of antimicrobial resistance, and maybe there would be more resources committed to techniques like bacteriophages to combat antimicrobial resistance?
I think the worsening crisis of MDR infections, along with the advanced technologies which are available for characterization of the viruses and the host interactions is prompting the re-evaluation of the phage therapy. And the second point is that the pharma industry, which has steered clear of the antibiotics, has been taking notice of this. So I feel in the coming years, more and more attention will be paid towards bacteriophages, as I said, because of the newer technologies which are available, where we can we can think about modifying the bacteriophage inventions as such. For example, till recent times, people are working with the phages which have been isolated from the environment, but now the genetic engineering and the system biology is available, where the phages can be designed. So there is a way that we can think of modifying the phages, and we can have them genetically engineered phages. Similarly, there are now means available where the genes responsible for interaction between phage and the bacterium can be identified. The delivery agents, as I've already said, are being worked out. So there are many advances which have been made and which are being made. So the future is quite bright, and people are working on these lines to make this therapy available as well as affordable for the masses. And I do see that now people will realize that there is always a threat. And as I said, it has been predicted that by 2050, this will be responsible for 10 million deaths annually all over the world. So that itself is an indication that people would like to curb this emerging antibiotic resistance among the bacteria, and the alternate strategies have to be worked out. And this is one, the bacteriophage therapy has got a lot of potential, and I'm very sure with time a lot of work will be carried out on these lines.
I hope that that is going to be what pans out, and as there is a greater focus on the problem around healthcare and the risk that microorganisms, especially when they're resistant to known treatments, the risk that they can cause at a global level. I hope that that will lead to a greater focus on bacteriophage research and more resources being committed. I think in that, the role of organizations which bring together all the field scientists, the healthcare community, the role of those kind of organizations will be very important. You are the president of one such organization, which is called the Society of Bacteriophage Research and Therapy. Could you please talk about the work that that society does?
Yes, since my focus is on the therapeutic use of bacteriophages, but let me tell you that now these bacteriophages are finding use in a lot of applications. They can be used as in agriculture, they can be used in aquaculture, these are being used in food industry for the safety, environment, and so on. So there are multiple scientists in our country who are working on different aspects of bacteriophages. And there was a conference on bacteriophages which was organized in Banaras Hindu University in 2017, and where all the phage scientists, majority of them, were collected. Dr. Gopal Nath was the organizing person. And there we felt that we need a place where we all can interact, and a society, this Society for Bacteriophage Research and Therapy, was then created, and we got it registered in 2018. And we basically have created a platform where the scientists, especially the young scientists who have just started working in this particular area, can come together, can interact with the senior scientists, can have interactions, can present or showcase their own work, and possibly it might lead to some collaborations as well. So the first conference of the a society was held at the Vellore Institute of Technology in December 2019, and it was very well attended by a number of people. The second thing which the society is going to do is to bring out the newsletter. And in this newsletter, the various activities which are being carried out, various scientists will be reported. The publications which are being made will be shared with the other scientists within the country. And if need be, tomorrow the society can also arrange certain workshops, etc., for the people who might be the beginners or something like this. So at the moment, this is all what the society is doing in terms of helping the newcomers, especially, to bringing them together so that the ideas can be shared and collaborations be made.
So encouraging the newcomers is a major role which the society is going to play. In your university and in your travel and lectures at various other universities like Vellore Institute of Technology, at your own university, do you see a growing interest amongst young scientists to get into the world of bacteriophage research and the applications of that?
Oh, yes. We have very young members of our society, and that shows that the young people are interested because, as I said, the bacteriophage therapy is one of the alternatives. There are other alternatives also which are being worked out, and the youngsters are very much getting into the research in these particular areas. They do see that there is hope where they would be able to bring out a product. But other than this, yes, we do see that the phages could be used in the agriculture, in the animal husbandry, the food industry, the aquaculture. A lot of research is going on in our country, and the youngsters are involved in this. As far as president of the society, I'm aware of this, that the younger generation is doing a lot of work in the area of bacteriophages.
That's really heartening to know. I'd like to talk a little bit about the global pandemic that we are all experiencing and living through. A lot of people who may not have known much about microorganisms, who were never really, in different walks of life, who never really knew much about viruses and bacteria. Everyone now in the world knows about coronavirus and COVID-19, and everyone is now read a little bit about viruses, and there is a big perception around how viruses can be very dangerous, which is we are seeing in terms of how the entire world is currently in a state of quarantine and restrictions of travel and going outside. Can you please talk about the difference between viruses like COVID-19, which are harmful to us, and viruses like lytic bacteriophages, which can be beneficial to us in combating our own health problems?
As far as coronavirus is concerned, it is a mammalian virus, whereas the bacteriophage is a bacterial virus. And we all know that the viruses, they cannot function or reproduce outside a cell, thereby being totally dependent on the host cell machinery for their replication. And they replicate once they enter inside the cell. And for the entry of, for example, for the bacteriophages, there are receptors which are present on the bacterial cell, which are not there on the mammalian cell. So the phage will always enter through the receptor inside the cell and then direct the host cell machinery to form the phages or the proteins for its own function. So there is a lot of difference. The mammalian viruses will attack only the mammalian cells but not the bacterial cells, and that is the reason that the phages, according to the FDA, they are safe and they are potent, and the non-lytic phages non-lysogenic phages can be very well used for the therapeutic purposes. So there is a lot of difference between bacteriophage and a mammalian virus in terms of its entry into the cell.
And that technicality makes the difference between whether a virus is potentially beneficial for us in, say, combating bacterial infections, or it's something that can impact us, which, say,
this is beneficial for us because it is helping us to destroy the bacteria. But for bacteria, it is not helpful. For bacteria, it is causing, if we are using a lytic, it is causing a lysis of the bacteria. So for bacteria, it is not helpful, but for us, it is helpful.
I can see a lot of questions are beginning to come in, sir. Before we get to those, I'd like to ask you one question about what you foresee as the future, what you envision as the future for phage therapy, both from the Indian context as well as the global context, say in the next 5 to 7 years. So in the year 2025, where would we stand vis-a-vis bacteriophage therapy, both in our country as well as across the world?
The study of the bacteriophages potentially throws light in the development of the new biotechnology products. And with my own experience in this area and the experience of the other scientists all over the world, that these bacteriophages are very efficacious in treating the bacterial infections in the animal models. So there is a hope, and I feel that there is a need now to turn the bacteriophage from a laboratory tool into an efficacious therapeutic for a broader market. And in this context, I would like to say that the companies are seeking to scale up the production and deliver potent phage products using the good manufacturing practices quickly and reliably. And many of the companies are working in this particular direction. And in fact, I would like to mention some of the companies which are already in this business: the Amphiphy Bioscience, the Biotics, the Phage International, the Fixed Phage, the Locus Bioscience, the Ferriseeds Pharma, Intralytics, Adaptive Phage Therapeutics, and Gangajan. Some of these companies are already in this business, and I'm very sure that they are working on various aspects of this bacteriophage therapy, and soon they will be able to come out with the technologies and the products, which again, as I say, they will be available for masses at a cheaper rate. One thing I would like to say is that the bacteriophages we are talking about as an alternate therapy. In fact, the antimicrobial resistance is so rampant now that this is happening. And with my own experience, I do get a lot of calls from the relatives of the patients in my own country, where people are looking for my help because their relative is infected with a drug-resistant strain and is not responding to the antibiotics. So in a way, I would like to say that this is happening, and therefore there is certainly a need to develop the alternate strategies, and bacteriophage therapy certainly has a future as far as treatment of drug-resistant infections is concerned.
That's a really good point that you make, sir, that it's a problem that we are facing today. It's not a problem of the future. It's a problem that we are facing today that we don't really have a solution to. So anything which does give us a solution, we need to fast track it and make sure that we can bring it to widespread use as quickly as possible, so that the maximum number of people who are suffering from these problems, to which there is no solution at this point of time, they can avail of this.
Yes, certainly.
I'd like to put a few questions to you, sir, that have come through this discussion. There are people who have posted questions for you. So Sumantro Chatterjee wants to know, that are bacteriophages more effective for external infections like skin infections, or are they equally effective for infections inside the body like UTI infections?
Um, yes, as far as the topical application is concerned, this is much easier. And therefore, for example, I have been using this in the burn wound infection model. But these can also be used as mentioned, that UTI. So they can be inserted into the bladder through the urethra using a catheter. And people have shown a lot of success as far as animal models are concerned. So UTI can be easily treated with bacteriophages by inserting the bacteria through the urethra into the bladder, and it works. And my own little experience, I also worked as far as the urinary tract was concerned. Yes, good success is there, but topical application still is much easier and is preferred with bacteriophages. But for other systems, some modifications have to be made before we can put them into the system as such.
So one is the modification to the phage that has to be made, and I think the second important factor is to get it till the site of the infection, because once phages reach, then they start multiplying. So
yeah, once the host is there, they will start multiplying. They will not start multiplying on their own, but if the specific host is there, then the important thing about bacteriophages is, vis-a-vis the antibiotics, is that antibiotic has to be given repeatedly. But in case of the bacteriophage therapy, if the host is available, they will multiply and the concentration will keep on increasing till the host is available within the system. So their concentration on the other end, it increases with time vis-a-vis the antibiotic, which decreases, and it has to be then given a second shot to maintain that particular concentration.
And since he had mentioned chronic UTI, I think the point you had made about bacterial biofilms also becomes important, that phage is able to puncture bacterial biofilms, which
yes.
Antibiotics are not able to
reach. Or even if they are able to reach, it is almost 1,000 times the more concentration as compared to the planktonic state. So this higher concentration then will be detrimental to the host system also. And therefore, phages, especially the depolymerase producing phages, can be very helpful in treating the chronic UTI infections.
Tripti Malik Ahuja would like to know, she says, please tell us about the possible application of phages in purification of water.
Purification of water is such, if you have, say, a particular bacterium which you want to target, then it is possible. But if you say you want to target all the bacteria which are present in the water, then you will have to first know that which are the bacteria which are present in the water, because the phages are specific for a particular bacterium. As I said, they have to have a receptor to enter inside the bacterium. So purification of water in general may not be possible, but yeah, if you want to inhibit the growth of Salmonella typhi, or if you want to inhibit the growth of Vibrio cholerae or Shigella, then you can have a specific phage which you can use, and you can get rid of the particular pathogen in the water system.
There's another question which is related, and I think the answer would also be related. Atif Khan would like to know that what are the possibility of application of bacteriophages for controlling biofilms in natural environments. He gives an example, for example, cooling water pipelines with continuous flow of water.
Yes, people have demonstrated efficacy of bacteriophages in destroying the biofilms in the pipelines, and there is a possibility, and this has to be worked out. Of course, you will have to first standardize the conditions as such, because they may not be very effective the way they are in the laboratory. So depending upon the area, depending upon the place which you want to disinfect or you want to treat, you will have to first standardize it first.
So I think that that is all the questions that we'll be able to take right now. Thank you so much, sir, for joining us and sharing your insights, sharing your experiences. This has been very valuable for everyone who's tuned in. And for everyone who's joined in, I'd like to thank you all for being a part of this, for sending your questions and comments. The information about our next session will be put out soon. So do stay tuned to our Facebook, Twitter, and Instagram to know when the next session will happen. Goodbye, everyone. Stay safe, and thank you, sir.